there were little dots that were on each of these. What actually happens is between this interface, protons can get here. They actually bind into one of those little dots, and it has to move around to find the channel out. And that's what's actually powering that. It comes in, binds into one of those pockets. As it moves, the proton gets moved over to another channel that goes in. So the channel for the protons is not straight through. It comes in and binds, gets moved over to the channel on the inside, and it goes in. The rotation requires 3 protons. So to go from one state to the next, it costs 3 protons to actually move through the channel. Okay, so 3 protons makes ATP. That's important. 3 protons through the synthase makes 1 ATP. Well, not APP. ATP. All right, so check this out. We know NADH, we said, pumps out 10. We know FADH2 pumps out 6. We know 2.5 ATPs are made with NADH, and we know 1.5 is made with FADH2. So if I take 10, divide it by 2.5, pull out your calculators. What is that? Oh, come on now. I don't see you actually doing that with your calculator. What number? 4. Okay. Do this. 6 divided by 1.5. Okay, pull out your calculators for real. How much do you make? What does that come out to be? 4. What did we just find from the experiment? How many protons needed to go through the pump to create 1 ATP? 3. Okay. By this calculation, it says that I need 4 protons to make 1 ATP, and it's pretty consistent. It's consistent from NADH and from FADH2. But the experiment said 3. So who is wrong? Or what is going on? It seems like we don't have perfect stoichiometry here. Okay? So what in the world is going on here? Is somebody wrong? Well, 3 is the correct number that has to go through the pump or through the synthase. And 4 protons are pumped out for every ATP made. And so there is some use of that energy, and here is the use of energy. All right, here is what's happened. I create lots of ATP on the inside. That's not where it needs to be used. It needs to be used in the cytosol. So what I do is I allow ATP to flow into a transporter, and it's transported out. Okay, so that's great. What happens is that ATP gets broken down into ADP plus PI. Now, for us to create ATP, I have to have ADP here. Well, if I keep sending this all out, my concentration of ADP becomes really high out here, and I have no ADP inside the cell. So what happens is I take ADP and put it in a transporter, which can go down the gradient and translocate back into ADP. But I don't need just ADP, I also need PI. And so I take PI, it goes the long way. Okay. And it will go in the cell, but what kind of transport is that? SIM port. And for every pi coming in, what has to come in? 1 proton. Here's where the fourth proton is used to create ATP. You use 3 here, and you use 1 to transport the pi. Now, what this slide doesn't do really well is this. They should show it. This is a new figure. That's a translocase. For every ATP that comes out, an ADP comes in. So that keeps the balance of ATP, ADP going. What isn't balanced is the pi. That's why it has to have energy come in with that proton to bring it inside the cell, or inside, sorry, the matrix. And so the ADP and pi are balanced.
How does the ADP go through that translocase transporter if it's closed like that? Does that have to do with the gradient?
Why does it go through? Oh, in terms of, yeah, it will go down the gradient for both of them. ATP will be much higher almost all the time inside the mitochondria, and ADP will be much, much higher on the outside at any moment in time. And so that's when the translocase starts. Now, if you get to a place where you're not using a lot of ATP, then those translocases will stop, and you will stop the whole production of ATP also. And so this will start working when you get those differentials. You get ATP really high inside and ADP really high outside. That's when it starts working. All right. So we can do all of this, but here's the problem. NADH, that is producing glycolysis. It's in the cytosol, but it must get to the mitochondria, and NADH can't be transported across the membrane. So it has to give its electrons to another species that can go through. There are 2 pathways, the glycerol 3-phosphate shuttle and the malate aspartate shuttle. The glycerol 3-phosphate shuttle is this. You take a little bit of the dihydroxyacetone of glycolysis, and you take this NADH that's produced in the cytosol, and you transfer the electrons to this ketone to create the alcohol. Okay? This molecule can't be transported. This molecule can't be transported, but this one can. There's a protein to allow it to go through. And it gets in, and it interacts with this dehydrogenase, and the glycerol gets converted back to dihydroxyacetone phosphate. But what I have done is I've taken my electron pair, an NADH, and I've transferred it to an FADH2. Here, I could get 2.5 possible. Here, I can only get 1.5. I've lost an ATP. And there are 2 produced in glycolysis. So if I have to put both of them this way, I lose 2 effective ATPs if I have to use this pathway of potential energy. Okay? That seems like a waste of energy. Okay? Now, the malate aspartate shuttle is a different thing. It occurs in the heart and the liver. Okay? And it allows us to shuttle the electrons into the mitochondria as NADH. And so what happens is the amino acid aspartate gets converted to oxaloacetate. Now this oxaloacetate here, we did produce oxaloacetate, but that was in citric acid cycle. That's down here. So this is not a citric acid cycle intermediate itself. The glycolysis produces the NADH, and it will reduce it to a molecule called malate. And malate has an interesting ability. It gets into an antiporter that puts in a malate, and every time it does, it will kick out an alpha ketoglutarate. this alpha-ketoglutarate is this reactant here. The malate then reacts and produces an NADH, creating oxaloacetate. And effectively what I've done is I've taken this NADH and I placed it here. I can react the oxaloacetate with glutamate, which is the reverse of this reaction, to make alpha-ketoglutarate and aspartate. And what's interesting is aspartate gets transported out, but a glutamate comes in, and that glutamate is that reactant. So they're interconnected. If I use this, I get all of the energy and I make 2 more ATPs. The problem with this is that this only works if this concentration is higher inside the matrix. Well, if the matrix is working, it's a little tail off. I don't have a lot of this. I actually have a lot of NADH. Only in a couple tissue types does it work out that this condition exists, and that was in the heart and the liver. In muscle, the shuttle, this shuttle here, even though it is more efficient in terms of extracting energy, doesn't happen very often. In all other tissues, the less efficient one works. And so when you go to the stoichiometries from ATP from glucose, you can create a potential of 3 or 5 ATPs from those NADHs, but most often you make 3. And so all the stoichiometry comes out, you can make 30 or 32. The maximum is 32, but most often you only extract 30 from glucose. And that's where the differential comes in. And so you can answer, if someone asked you how many ATP can be made, the answer is the maximum is 32, made in only certain tissue types. The most often is 30, made in almost all tissue types. So it's a conditional answer. All right. That proton gradient, we're burning fuel, okay? We convert the energy of the glucose or all the fuels that we eat into a proton gradient, and then we can convert it back into ATP. That is one type of energy, all right? There is one additional energy that you can create through this process, and that energy is heat. And that's important. Now, this was a diet drug that was given for just a little while until we found out it was really, really dangerous. Okay, 2,4-dinitrophenol. 2,4-dinitrophenol. You can go into the organic stock room, find a bottle of 2,4-dinitrophenol. Take a spoonful of it, right? Is that what you're going to do? No. Okay, so this is what that particular drug did. It has the ability, in this space, it can pick up a proton. In this form, the protonated form, in the protonated form, it's hydrophobic. Hydrophobic enough to go through the membrane without a transporter. Gets in here, it can deprotonate, okay? And actually, this can, even in the ionic form, it can go through the membrane again also. And so what this does, it doesn't go back in. It can transport one each time, and this builds up inside the mitochondria. That was one of the bad things. But what happens is, if I take this drug and it puts protons inside, what I've done is I allowed the breaking of that proton gradient, and I get no ATP. Okay? The body still needs ATP. So it's still going to burn sugars, and once it's done with sugars, it's going to start to burn fat, and it'll burn fat, and it'll burn fat. And I don't make any, or I make less ATP than I did. Okay, worked pretty well. The only consequence was people got hot. Okay, but there's a balance with use of this drug. Okay, if you have too much of this, then all of the gradient will flow this way, and you will make no ATP. Not a good drug. Okay, because if you don't have ATP, you don't live. But here's the consequence with people who are taking medications to lose weight. Well, if one pill works good and I'm losing lots of weight, what if I want to lose more weight? Take 2 pills, or I take 3 pills, or I take 4 pills. I'll lose 4 times the amount of weight with 4 pills, right? No, you just stop making ATP. You're dead. Okay, and so this was an idea that just didn't really work out too well. Okay, but there is a natural process. It happens in hibernating animals and in newborns. It is in the form, and this process happens in what's called the brown fat. If you actually look at adipose tissue under a microscope, the actual cells, they look translucent to white. But in hibernating animals and in babies, you don't see it from the outside, but the baby fat, some of that is brown adipose tissue. The reason why it's brown is because it has so many mitochondria inside of it, it actually changes the color. And those particular cell types have lots of mitochondria, and they have one protein that is upregulated called thermogen. It's also called the uncoupling protein. And what the uncoupling protein does is it embeds into the membrane of the mitochondria. You break down lots of sugar, lots of fat, lots of fuel, and you pump out protons. And what it does is it competes with the ATP synthesis, but it's regulated so that it will never overtake the ATP synthesis. And it will allow some of the protons to flow back through. The only thing that you generate, the energy that is generated, is converted into heat. And so in hibernating animals, this keeps their body temperature. And in newborns, they are so small that they need this extra generation of heat because they lose heat faster than we do. And so during infant stage, the baby fat will eventually burn off and you will lose the brown fat. But it's temporarily with infants, so until they get large enough that the loss of heat, just because of size and mass, isn't a large percentage of their loss of heat.